Hello, this is Ahmed Al-Hulayl. I will represent my co-authors by presenting a summary of our work that study the feasibility and reliability of our accelerated renal MRSI technique, which we believe can be a promising tool to investigate heterogeneous renal cell carcinoma. I have nothing to disclose. The renal cell carcinoma are characterized by being a lipid-rich. Its differentiation from other histological subtypes could be feasible by the detection of intracellular lipid contents. Thus, a method to differentiate intracellular lipid contents is needed. Talking about the common methods of human fat measurement, we have the in vitro method, which is invasive and not suitable for follow-up studies. It also provides no information about the spatial distribution of lipids. It represents only a small renal volume, which makes it not a good choice for heterogeneous RCC. On the other hand, we have the imaging method. It maps the summed fat fraction, so it doesn't provide information about the different lipid components. To solve that, we usually use the single proton MRS, which can differentiate the intracellular lipid, and its feasibility has been reported in healthy volunteers and patients with RCC. However, it cannot map the lipid distribution within heterogeneous tumors. So, the conventional multivoxel MRS imaging can deal with the heterogeneous tumors. However, it requires a long acquisition duration, ranging between 15 to 60 minutes. So, the goal of this work is to investigate the feasibility of our fast metabolite cycled, density-weighted, concentric ring case-based trajectory MRSI acquisition to acquire high-quality quantitative renal data in healthy volunteers within a short scan time, about 3 minutes, with the intent to establish the signature of the lipid composition of healthy renal tissues, which will be used as a future tool for non-invasive characterization of RCC. For more technical details about the sequence, I would refer you to our previous publications, which can be reached by scanning the QR on the right. In our study, we used a flexible 18-channel body coil attached to a 3T Siemens Prisma scanner. The coil was used to acquire abdominal test retest scans on 5 healthy human subjects. Between the test and retest scans, the subjects took 5 minutes break outside the scanner. For the MRSI scanning parameters, a 48 by 48 voxel was assigned, where each voxel has 0.25 milli-resolution. The study performed with one average and without implementing respiratory navigation or triggering. For lipid quantification, lipid peak signal were fitted by using LC model software before the fat fraction was calculated using the shown conventional fat fraction equation. To assess the resulted MRSI structural maps, they were compared with their corresponding high-resolution MRI images. Beside the differences in resolution and contrast, where the MRI image is T2 weighted and MRSI is proton density weighted, there is an agreement in anatomical distribution between the MRSI and the MRI image. Here the yellow box added to show the shimming area covering the left kidney. Here is a representative data showing the image acquired during the baseline and repeat scans. To give a better insight, the fat fraction maps calculated from the MRSI data were overlapped on the corresponding structural MRI image. During the planning, in order to help in the repeatability assessment, the helium of the kidney was used as internal landmark within the slice. On the image on the left, an example of the region of interest that was used to evaluate the MRSI quantification repeatability. The region of interest was carefully drawn over several MRSI voxels covering only the cortex of the kidney to reduce the anatomical variations. The repeatability results were summarized in the table. The fat fraction percent was ranging between 1 to 2% between subjects. The intra-subject coefficient of variation was always less than 10%, which reflects an excellent repeatability. To show the level of repeatability of the signature of lipid composition, a lipid spectra from the cortex of the kidney that acquired from the baseline and repeat scans were averaged. The black line represents the mean, while the brown shade represents the standard deviation. The blue box highlights the lipid peaks differentiated by the MRSI method. In conclusion, we showed that the proposed MRSI method has several advantages. It provides the content and spatial information of kidney lipids with high in-plane spatial resolution, 5 by 5 mm squared, which is higher than most common MRSI techniques. Nevertheless, the data can be acquired within a short and clinically acceptable scan time. It also showed a high degree of reliability, which supports the future application of this technique for non-invasive characterization of heterogeneous RCC. The limitation of this sequence is lower spatial resolution compared to the MRI techniques. Here, I would like to thank you, my collaborators, my co-authors, and financial supporters.